Hello everyone and welcome to my webinar on engineering membraneless organelles step towards cell-free paranoids. I am Gaurav Chauhan. I am an assistant professor in the Department of Chemical Engineering at IIT Indore, and this work is going to be done by the generous support from ANRF, and I would also like to thank IIT Mandi for this opportunity to participate in the national webinar competition. Now, my lab works on understanding how phase behavior can modulate compartmentalization inside cells. So, as we know that most of the compartments inside cells, something that we find in our textbooks, are these organelles. And so an example of an organelle would be nucleus, it would be, Golgi bodies, it would be a lot of these membrane bound compartments that we call organelles, and essentially membranes are what provide the necessary compartmentalization, and they control which macromolecules or molecules are going to be present in which compartments, right? Now, in the recent times, some of the interesting work has led to the discovery that we need not have membrane bound compartments, and in fact we can have a spatial organization or compartments which can come about because of phase separation. And here scientists have discovered a lot of phase separated compartments such as nucleolus, Cajal body, paraspecle, nucleus speckle, stress granule, germ granule, P granule, and so on and so forth. And the idea is that these compartments can come about due to phase separation. So just like how we know that oil and water can phase separate, in this figure oil and vinegar phase separate, right? So as olive oil and vinegar phase separate in this nice little photograph, what we can see is that droplets come about. And the thing to notice about these droplets is that the droplets are enriched in one component and they are deficient in the other, right? And in the seminal work carried out by Cliff Brangwyn in Tony Hyman's lab, what they have observed were these P granules which looked very much like oil and vinegar droplets, right? And so this is the idea that phase separation can result in the formation of these membraneless droplets which can compartmentalize the cellular environment. And much like how chemical engineers study about upper critical solution temperature or lower critical solution temperature, as the researchers have cycled through temperature, what they can see is that these droplets can appear and they can dissolve, right? And which shows that there is a temperature dependent phase behavior for these droplets. Now, one of the compartments that we are interested in, which come about because of phase separation, are these pyrenoids. And pyrenoids, for people that don't know about pyrenoids, these are essentially the compartments inside the algal chloroplasts which are responsible for fixing carbon dioxide. And this is particularly important when we think about engineering better pyrenoids, because that could mitigate the carbon crisis that we currently know that we have. And so in this pyrenoid, the pyrenoid is also a phase separated compartment, and it is formed as a result of phase separation of Rupisco enzyme with its scaffold protein, which is known as EPYC1. And so the question that our lab is interested in asking is, what is the functional consequence of having this pyrenoid assembly be a phase separated assembly? And so this is the question that our lab is focusing on in this project, which is how are enzymatic reactions going to be affected by phase separation? And here we are going to use kinetic Monte Carlo simulations to study reaction kinetics in the two phase systems, and essentially generate a rule book or a guide to show where in the phase diagram should we operate in order to have maximum reaction efficiency. Now, another thing that we are going to do in this project is to generate phase diagram of pyrenoid. And so this is what a phase diagram look like, which is essentially going to tell us what is the concentration in which we are not going to see a two-phase system or droplets, and in which concentrations are we going to see two-phase systems. And essentially we want to use computational modeling to predict the phase diagram of pyrenoid. And then second thing that we want to do is essentially ask, okay, if we do have droplets, how are these droplets going to modulate the reaction rates, right? And sort of how the computational engine works is that we start with a protein system where we define the initial structure, and then these are essentially points on grid, right? And then we can define what are the interaction between different points, and that is what we call as force fields. And then what we are doing is we are just solving the Newton's equation of motion as a function of time, and then we get this trajectory which we can analyze. Right? And so in the end, what we are going to get out of this project is we are going to have prediction for phase diagram for the pyrenoid assembly. We are going to know, okay, under which regime should we expect the enzymatic activity to be enhanced. And then finally, this is going to lay the groundwork for design rules for effective pyrenoid assembly that we can essentially get in a cell-free setup. Thank you for your time, and thank you to ANRF and all my colleagues and lab members. Thank you.